底定的就是在香港。那未来我们也可以到我们新加坡总部这边去拍。所以今天我们特别请到说那个李天益博士帮我们招一下手好吗？李天益博士and then we have a application team from Groningen that is Gero. Can you please wave? Thank you. So in the first beginning, we are going to have a 30 minutes, but it is by Mandarin. So it's 30 minutes. Dr. Tian-Yi will let us know what is the basic concept briefly, but 对不起，我们侧重的还是在 application 上面。那么在整个过程中，我们会录影，到时候如果说有老师来不及的话，我们会放上连结跟各位联络。那我们就先请天益博士帮我们讲一下，就是有关这一次STED 的那个machine， 它叫 CT line。在天益开始之前，我先跟大家提一下哈，因为今天是2个小时，那我们没有特别设中断时段。那如果说有需要有什么问题的话，可以跟那个 Neil 联络 ，Neil 大家都认识，然后或者跟我联络，我们会一个一个跟大家说明清楚。那会后如果要资料也都可以跟我们联络。那特别在做 3D 的时候，因为 3D 是不止在 XY 平面，还有 Z 平面各这个轴向的这些要考虑到的地方，那天益博士会跟大家解释。好，我们现在就请天益博士。好，天益麻烦你哦。
好的，感谢 George 的介绍啊。很高兴今天有这样一个机会跟大家介绍我们 Abberior 的超分辨系统。那么我是负责亚太区的这个 application scientist。
天益你 mute， 天益你 mute。
喂，现在大家能听见吗 ？OK，
可以了，可以
了。不好意思，那么我前面以这张 slides 开始，就是这是一个非常好的杂志，在2017年做的一篇 review， 里边列出的是不同的这个 light microscopy 它的 resolution， 就是说PSF 点扩散函数。如果大家对这个概念不是很清楚，我们就可以想象你有一个无限小的点，那么在不同的显微镜底下去成像，那么最后得到的结果它的大小，这个图像越小，就证明它的分辨率越高。那么像 confocal 的话，Z 方向都要500-600纳米 ，XY 方向是200-300纳米。像 SIM 呀，大家听说的，还有 Ariscan， 它这样带螺纹的是做了一个这个 deconvolution， 就是内部的一个计算。上面是常规的，那么底下呢，像大家听过 STED、PALM， 这是2014年拿过诺贝尔奖的技术，它们是一个传统的超分辨技术。那么 STED 大家可以看到，它是不通过计算的，所以它是物理直出型的这种超分辨系统。那么 PALM 呢是通过计算，然后相对来说，它们2个的分辨率是可以比较的，然后。相对更新的是2017年由 Stemhell 教授提出的 MinFlux， 然后这个分辨率大概是1到3个纳米啊，让大家有一个这样的一个感觉。整个这个 PSF 现在是一个 X 轴和 Z 轴方向这样去做一个展示。那么如果我们现在往下看，就是说我从 XY 方向给大家展示，那么 confocal 刚才我说了，它的这个 XY 方向是200到300纳米，根据你用的波长不一样，分辨率有一个区别。那么我们 Abberior 公司的 STED 系统大概是20到40个纳米的分辨率，还有就是 MinFlux 系统是1到3个纳米的这个分辨率 ，PSF 的对比是非常明显的。那么后2个 STED 技术和 MinFlux 技术都是由 Stemhell 教授发明的。那么他因为上面这个技术得了诺奖，然后这个是得了诺奖之后研发出来的技术。然后这2个技术实际上我们现在都是给它成功的作为这个商业化了。我们的机型大概有些老师已经看过，就是有这个 STEDYCon 在台湾这边做了 demo， 它的分辨率大概是30到40，是一个非常小型化，我们叫入门级的 STED。那么它大概主机就只有一个电脑 PC 那么大。然后呢，我们还有这种大型的一个叫 Facility Line， 还有一个叫 Infinity Line。Facility Line 是一个标准化的一个高端机型，那么 Infinity Line 我们可以做定制化的。那么这2个系统都是我们经过多年的迭代，除了最基本的 STED 功能，现在呢又加入了3D STED 以及更多的高端的这些特色。一会儿我会一一的给大家 做一个介绍。那么而且这个高端的，因为它功率比较高，所以 XY 的分辨率也是比 STEDYCon 要高，是20到30，而且这方向是显著的升高。这个 Z 是500个纳米啊，跟 confocal 一样。然后除此之外，我刚才说了，我们Abberior公司有 MinFlux， 这个分辨率就厉害了，1到3。然后 XYZ 3D 的话可以做到3到5纳米，这是世界上目前商业化光学显微镜里分辨率最高的。好，那么我们今天重点是在说 STED， 我简单的过一下在 STED 的原理。那么 STED 实际上是基于 confocal 搭建的一个系统。那么 confocal 大家都了解，有一个激发光通过 lens 之后，你拿到一个在这个 focal plane 可以看到这样一个scanning spot。那么刚才说了，你无限小的一个点，最终因为光学衍射极限的存在，所以它总归这个点最小也是200-300纳米。然后在这个区域，如果我们放大的话，里边有好多，比如说荧光分子，我们给它去做这个激发，然后最后它从这个高能态下来会发射出它应该有的那个荧光，这就是我们大家看到的。但是实际上这个点是非常大的，里边的细节你是看不到。这就是为什么 confocal 它受光线衍射极限分辨率，导致我们一些精细的结构不能够解析出来。那么 STED 是怎么样呢？就是我们在原有的 confocal 基础上加了一个 STED 激光，然后这个激光是什么样的 pattern 呢？是这样一个多纳圈的形状。那么这个多纳圈和我们的 excitation laser 它是同心的，是完美的耦合到一起。那么在这个时候呢，就是说还是我们把这个荧光分子就是外圈，大家看外圈如果有荧光分子的话，还是由低能态激发到高能态，但这时候我加了一个 STED 激光，给它强行用高能态压下来，这时候它的荧光分子就不发它本来原有的那个荧光了，它发 STED 激光，然后我们再拿一个滤片把它滤掉，所以感官上外面就 switch off 掉了，你就看不见它了，只能看见中心点，所以最后看下来，它的照明区非常小，所以就能解析出非常好的、非常这个分辨率非常高的这样一个结构。正因为外面用的是 STED 激光，所以这个激光的强度是可调的，所以整个系统它又是一台 confocal， 又是一台 STED， 而且这个 STED 的分辨率是可调的。我们看，这是一台这个 facility 拍摄的，我们就把它只开 confocal 模式，它正常的一个20纳米的小球拍出来200多纳米，对不对？那么我们加一点 STED 激光上去，功率提高一点，这个分辨率就有一定的提升。不断的加 STED 激光的功率，最后它一直到它本有的大小，大概20纳米的这个粒子这种情况。那你要是这时候把 STED 激光再调小呢，它又回去了，是这么一个情况。好，那首先说一下这个 STEDYCon，有的老师可能已经见过这台机器了。刚才说了，非常的紧凑、小型化，但是它的配置一点也不低端，整个用的激光全部都是 pulse laser， 然后有这个高端的 detector 和这个镇静。那么简单稍微过一下这个，就是说我们要想做一个非常好的STED 的结果的话，其实需要各个硬件去完美的去匹配，所以我们整个的这个系统是全脉冲匹配的 excitation 完了之后，然后 fluorescent 出来，在这2纳秒之内，然后马上打一个 STED 激光啊，效调的非常好。那么传统的 STED 的话，它是 CW 的，它一直在开，这样是容易淬灭的。那么我们在小型化的这个 STEDYCon 上就不存在这样的问题。然后用的检测器呢，大家可能有的人也听说过，是 APD 检测器 Avalanche 的 photodiode， 这是水泵二极管，然后它的这个 sensitive 是非常高的，远比我们常见的这种hybrid detector 啊或者 PMT 的这个sensitive 也高得多。然后我们用的镇静是4个镇静，一般的 confocal 大家平时用的就是有一个 X， 有一个 Y， 这样它可以从上面扫下来就够了。那么有的呢，他们是用了3个镇静，我们 Abberior 是独家用4个镇静的，所以解析能力也是非常的强，能够保证这个多纳圈一直是完美的，所以实现一个非常好的这个 STED 效果。那么看一下我们的这个结果，这是 STEDYCon 小型机拍出来的，这是拍的一个 centroid， 在 confocal 模式下就是2个点，对不对？那么如果我们开了 STED 模式，就能够看到这2个环，而且这环里边的亚基是能够看出来的。这个时候我们可以做一下 deconvolution， 然后把这个结果进一步的这个锐化 ，contrast 得好，然后分辨率也更高。所以这里边我要说一个概念是什么呢？就是 STED 它是一个光学直出，我们本身这个结果其实可以做 deconvolution， 也可以不做，你直接用这个结果也可以。 而且在这种情况下，因为你已经看到了各个压机，所以你即使用 deconvolution 用算法去算这个结果，你心里是很踏实的，你不怕它算错，因为这里边其实结构已经出来了。然后这个你就看起来很幸福，但是大家也知道，可能比如说用过 SIM 啊或者一些其他的这种成像技术，它靠很高的算力，在这面可能还是一个点，连圈都看不出来，然后这面算出一个圈或者算出一个压机来，这个时候其实大家拍的时候心里是不踏实的。已知的结构用算法当然是很容易算出来，但是未知的结构你不知道它算的是对不对，这就是 STED 整个这个技术的优势，它是一个物理直出型的技术。我们的系统输出的都是直出这样的一个图像，原始图像没有经过任何的这个 post processing。然后这是另外一个结果。那么我们用 confocal 拍的话，看这里一个点是吧？其实它是 vesicle 标的是 cholesterol 网格蛋白包裹着那个 vesicle。用我们的 STED 模式一拍，可以发现哦，这个其实大的点不是这样的，它是一个中空的结构，真的是一个 vesicle。正面其实还不太好说，正面网格蛋白其实它没有进到 vesicle 里边。然后这个呢是一个我们这边在北大的一位老师他自己设计的一个线粒体染料，大家可以清楚的看到，这拍活细胞能够看到这里边线粒体的脊非常的清楚。简单说了一下 STEDYCon， 现在我们开始最重要的说 facility line。那么 facility line 具备全部刚才我说的 STEDYCon 的那些特点啊，它是用的 pulse laser 啊，然后它是 APD 探测器啊，还有振镜也而且它的这个激光功率要比 STEDYCon 要高得多，所以它的分辨率本身也会高。刚才说了对不对 ？STED 激光的功率越高，开得越大，它的分辨率越高，所以它的分辨率会提高，而且它是能够做3D STED，所以 Z 方向的分辨率也有一个提高。那么除了这些最基本的功能之外，其实它加了很多模块，还有这些更高大上的这些特点。那么我一一给大家做介绍。首先说一下 3D STED， 我相信这也是大家最关心的。那么什么是 3D STED 呢？首先刚才给大家看过这张图。就是说，我们2D STED 在 XY 方向是这样的，就是一个20纳米的这个小球，然后我给它加 STED， 分辨率就有一个提升。但是 Z 方向是什么样子呢？我们现在把这个样品给它翻过来看 XZ 方向。XZ 方向其实 confocal 的话，就是刚才说的那个大椭圆，对不对？你加了 STED 激光，2D STED， 它在 XY 方向的分辨率提升了，但是在 Z 方向，你看长度还是一样，并没有什么提升。对不对？它由原来的这个椭圆形变成了针形。那么如果把这样的样品，这种荧光粒子放到我们的 facility 来或者 infinity 来这种高端的3D STED 系统上，它才真正的能拍出它本有小球的那个样子。所以说我们的激光功率像这个开得非常高，那么就是一个点，一个小球 ，XY 方向各项同性都可以实现这样一个高分辨率的成像。这就给大家介绍一下这个 3D STED 能做什么。那么它是怎么实现的呢？从原理上，大家对这个多纳圈肯定是比较熟悉，对不对？刚才我也提了，就是想实现一个STED，一定要有一个多纳圈这样的激光，这个是从 XY 方向讲。那么从侧面看 ，XZ 方向，实际上这个多纳圈激光是这样的，是这两坨，它是一个立体的啊，不止这两坨，是一个筒子样的一个光。那么如果我们现在看 confocal 的 PSF 是这样的，我们把这个光套到这个 confocal 上面，就实现了这样一个针形的最后一个有效照明区。所以它的分辨率最后就是 XY 方向25纳米，但是 Z 方向跟 confocal 一样，这个就是 STEDYCon， 但是它功率没这么高，它只能30到40纳米。我们的这个 facility 下来能达到25纳米。那么我们要想实现一个 3D STED 呢，实际上不只需要 XY 方向的多纳圈，还需要 Z 方向的。Z 方向也要有 STED 激光上下。那我们把这两个光放到一起，就会实现这样一个光，就是整个它把 confocal 的这个 PSF 用 STED 激光给包围了，包起来。那么这两个图如果 overlap 的话，最后就得到这样一个非常小的有效照明区。所以说，XYZ 各项同性能够实现75纳米的这个分辨率。那你说为什么不能实现25纳米啊？25纳米，因为你把很多功率其实分布到 Z 方向了，整个我们的激光，分布到这面之后呢 ，XY 方向也有一定的这个缺失，所以为了保证各项同性是非常重要的，我们最后做重构非常重要，所以就实现一个就是各个方向，分辨率都是一个适中，然后适合我们做3D 重建的啊，基本它们对比是这样一个分辨率的 最大，然后变成针形在一个小点。然后这个事情操作复杂吗？对于软件来说，操作非常的简单。实际上就是我们这有一个 slider， 那么2D 模式它在这面。如果我们现在从2D 就是这个光路模式转换成这个光路模式，就把这个 slider 从这头拉过去，马上就转过来了。其他的操作根本不需要其他变化，你点一下 scan， 然后拍出来的就是 3D 效果。而且我们的系统有一个特殊点，就是这中间是无极变换的。我刚才说了，你的光分到哪里，哪里的分辨率就高。那有的人说我这一方向需要分辨率高一点，但不需要那么高，但是 XY 我还也需要的分辨率还比较高，那你就不要把光分到 Z 那么多啊 ，XY 分的少一点。所以这中间无极调节，全世界目前只有我们能做，是这样的。那么这是怎么实现呢？实际上我们用的是一个Spatial Light Modulator。那么通过这个空间光调制器，我们把一束 STED 激光进行调制，又出现这个 XY 方向的多纳圈 ，Z 方向呢也有这个光。这是我上了不同的维彩，但实际上这个光斑是从这一个 STED 激光里出来调制出来的。也就是说，它原来只是一束光，现在我调制出这样一个光斑啊，这是通过 Spatial Light Modulator。但是传统的，比如说像徕卡，他们做了10多年的这个STED 技术，他们其实还用最原始的办法，就是我把 STED 激光分成2路，那么一路呢，我给它形成一个多纳圈，另外一路呢，我形成这样像糖葫芦一样，上下有那个Z 方向有那个STED 激光，然后我再把这2个光给它做这个 realignment， 要让它2个合到一起，合完了之后就出现我刚才说那个 3D STED 的那个效果。但是这种最传统的这个老的技术，它有一个问题，就是它2个光的稳定性很差，你稍微一偏，它就做不了 3D STED 了。所以说，工程师刚装完是这个样子，但是过了1周，过了2周，大家可以问一下，用过其他系统的这个老师们，是不是 3D STED 平时是做起来很费劲的？那么我们因为是一束光，天然的是一束光，所以它不管怎么样，这2个这个 Z 方向和这个 XY 方向是一直稳定的在一起的啊，这是一个巨大的不同。所以说这种是更稳定的。那我们看一下实拍的效果，这是一个细胞核啊，有上层下层。那么我们如果用普通的2D STED 去拍的话呢，只能出现这种针形。XY 方向，如果你从 Top View 上看，肯定分辨率高了，但是现在是 XZ 方向不行。那么换了用 Facility、Infinity 这种 3D 模式去拍，清清楚楚的看到有上面一层。下面一层，而且里边每一个像这个拉明和 NUP 153都能解析出它本有的那个大小，更接近它本有的这个大小，对不对？这样您可以想一下，做这个 3D 重构的话，这面是完全做 3D 重构，这一分辨率是不行的，这面就会非常的漂亮。然后这是一个更极致的这个解析的这么一个例子，就是刚才那个细胞核的核膜，那么它上面有这个核孔复合物，那么核孔复合物实际上是核膜外和核膜内都有一个结构的。我里带的解析出来，你看上面有一点，下面有个点，上面有个点，下面有个点，这是通过非常高的 Z 轴的分辨率而实现的。这是另外一个，这是重构的一个效果。这是在 T 数里边拍这个 Brain Slice 里的这个 dendrite， 那么 confocal， 然后加这个。跟这个 STED 3D STED 的一个对比，你发现无论它怎么转，各项同性的这个分辨率都非常高，而且这里边还提出了一个叫 diming， 那么我下面也会介绍它到底为什么要加了一个 diming 这个东西。那么 diming 实际上就是我们另外一个特色，叫这个 flex exposure illumination， 或者我们叫 adaptive illumination， 是一个光保护的这么一个机制。那么这个机制是什么样呢？我放一个这样的一个动画。就是我们有一个 STED 激光，中间是激发光。当我们拍样品的时候呢，这是一个荧光分子，它就一直开着。这 STED 激光一直开着，刚才说了 ，STED 激光功率很高，功率越高，分辨率越高。你让它一直开着，总归这个光漂白是比较厉害的，对不对？那么我们有采取一个什么样的这个方案呢？就是说，传统的是这样一直开着，但我们现在是研发了一套这个智能系统。只有它遇到荧光分子，它才自动的把 STED 打开，没有了它就关上，有了它就打开。有了这样一套机制，那么整体你对这个样品单位面积产生的光读性和光漂白 都大大的减少。所以我们看一下，我们内部做了测试。那么对于传统的 STED 的话，它的光漂白非常严重。那么即使是用 pulse laser 还是比较高。那么我们现在用了这个 rescue， 其实是这个adaptive illumination 的其中一个点，它有不同的这个模块，加了一个模块都可以减少这么多啊，是它最传统 STED 技术的1/25，而且跟 Palm Storm 这种 super resolution 它的光漂白相比也低。那么用下来是一个什么效果呢？就是说我们如果拍超分辨连续拍，那么我们用了 adaptive illumination 的时候，就可以多拍很多 frame。底下呢就不可以，因为当你用特别高的 STED 激光，我们就追求极致的分辨率的时候，就会出现这样的现象。那你加了这个模块，就可以拍更多张，这对活细胞呀，对 Z-stack 都是有好处的。所以说拍单个平面这是有好处的。那么 Z-stack 就像我刚才说的，这是核孔复合物染的细胞核，我们分别拍2个区域。左边这个区域拍的 Z-stack confocal， 这是 Z-stack 是用的 3D STED 拍的，然后加了 dimming， 就是 adaptive illumination， 我们可以看到它分辨率非常好。然后右边呢也是拍一个类似的效果，但是没有加 dimming， 拍完之后右边就脆灭了，左边这还完好。那么这就告诉大家什么呢？说如果你没有这个模块，首先是样品就光漂白是比较厉害，还有一个就是说左边的这个区域，其实我们还可以进一步的怎么样增大 STED 的功率，可以分辨率比右边还要高，所以这非常有用。好，接下来呢，就跟大家介绍另外一个特色，就是我们还可以做深层组织的 3D STED， 这个也是目前只有 Abberior 可以做的，我们管它叫 reshape。那我们知道，其实在拍样本的时候，就 confocal 还好，还是普通的观场，总归有这种optical 的这个 aberration， 对不对？就是你希望这个图像非常的 sharp， 但实际上有各种的光学畸变，越深这种光学畸变越厉害，所以你拍一些复杂的样本是非常难拍的，就是像这样的图。那么这个有可能是样本的事情，有可能是这个我们制备的时候 mounting medium 用错了等等一系列的这个原因。那平时我们说一个光打过来，然后正常它可以对焦，对不对？但是你有个样本，它里边的折射率非常复杂的时候，这个光斑的这个焦点就变了。所以你看这样一个图像，这是 XZ 方向，那么到底下越深的地方，因为光学畸变导致你这个图像就模糊了。那我们的方案是什么呢？我们加入了一个模块叫 deformable mirror。可变形镜，这个变形镜实际上是天文上用的，它怎么工作呢？就是它上面的这个表面是可以动的，可以做出这个校正。那么我们看，如果做出校正之后，它原有的那种畸变的那个 focus 就回到它本该有的那个位置，所以说最后你在收光子的时候能收的信号比原来更好，所以这时候上下的明暗度就比原来好得多，对不对？它整个的 contrast 的分辨率都能提升上来。这只是 confocal， 但做 STED 也是这样。我们这个 Drosophila 是一个比较大的样本，然后我们看这2个区域，最后拍下来，如果没有这个自适应光学的话，这个图像是模糊，然后分辨率不好的。那么右面加了以后，马上这里边的细微的结构在15个 micron 的地方就已经有这么大的区别了。其实更深的话，区别会更大。那么接下来是另外一个样品，就是我们现在有一个渗片，然后这个渗片再用普通的 3D STED 去拍的话，那么前几十个micron 就10到20，这个一般大家拍 confocal 拍的都知道，拍1、2个细胞的厚度是没有问题的，但是你拍深层组织，这个信号会随着这个深度的增加越来越弱，越来越弱，对不对？你几乎不可能拍这么深的地方，你只能说用这种2 photon 啊或者其他的技术，但分辨率就跟这个超分辨没法比了。那我们现在加了这个自适应光学这个模块之后，这个 reshape 的模式，发现上面跟下面这个信噪比都有一个非常大的改善。大家可以看到这个深度接近100个 micron， 然后这个是跟样品有关系的，有边制备好，其实比这个还要做得更深。所以我们现在是可以在很深的地方实现一个 3D STED 的这么一个好的效果，而这个效果不是说只能用于 STED， 刚才也给大家看了 confocal 又会非常好的这种校准。这有一个总结，就是说我们拿一个荧光小球做了这个篦子，然后比较深啊，这是 Z 方向，那么表面看的篦子是这样，深度的话就几乎看不清了，就任何的 confocal 显微镜拿这个样品一拍，那底下都是这样。那我们在 confocal 模式我就加了 adaptive optics， 底下你看这个信噪比提升了一大块，对不对？ 然后我们再用 3D STED 去拍的话，如果没有这个校准，没有这个校准，是不是底下就糊糊的？加了这个校准，哎，底下的分辨率就跟上面接近差不多了。这是一个很深，你看底下的 bar 是5个 micron，这是几十个 micron 的深度，啊，这是一个大家总结一下，到底这几个技术结合到一起有什么样的效果？那么最后一个呢，我还加了一个叫 matrix 的东西，大家看是不是线道比更好了？它是什么作用呢？是去背景。那我再说一下 matrix，matrix 也是我们 facility line 上有的一个特殊的这么一个功能，它是一个阵列型的探测器，有点像 iris scan， 但是不是 iris scan， 因为 iris scan 用的是 PMT 检测器，我们这边用的什么 APD， 就是更 sensitive 的，是能够做单光子计数的这样的一个探测器。那么它就像这个昆虫的复眼一样，怎么说呢？这是个阵列的探测器，那么可以从不同的角度去看同一个样本，这样它就能够从物理上区分到底哪个是背景，哪个是前景，哪个是你要的信号，然后物理上去背景。当然也是需要计算，但是实际上是通过物理不同的角度去做这样的一个计算的。我们看一下实际的效果，这是 Caco cell， 就是这个小肠绒毛的那种细胞。那我们现在在这个上面想做 STED 是非常困难的，因为它表面非常的复杂，非常的厚。那么你做 STED 的话，你很难找到非常完美的 focus， 看起来很多地方都糊糊的。但是我们如果在这种情况下换用 matrix 探测器去拍的话，整体背景就去掉了。然后你这个图像就看起来非常的通透，对不对？分辨率其实也有一个提升的。那我们再看下一个，就是还是这个图，但是我们做了3D 重构。开始这是传统的 STED， 就是不加 matrix 的，这个我们加了 matrix， 效果就有一个非常好的一个重现。这个是根据样品，不是说每个样品你普通的 STED 都拍不了，是有的样品像这么复杂的，有几十个这个10、20 micron 这么厚的一个样品，表面是需要 matrix 去给它去背景的。我们根据不同的这个样品做了这样的一个优化。那实际上呢 ，matrix 它的算力怎么样？还非常的高。它不仅是可以做单光子计数，然后它计数完还可以做一个统计。那么这就可以实现什么 fluorescent lifetime。所以我们说这个功能叫 timable， 其实就是我们平时知道的那个 flame 功能啊，荧光寿命。所以 matrix 可以做这个flame。比如说这是我们那个工作界面，就是我们显微镜它的工作界面非常的简单。平时我们拿这个样品过来，如果就是一个光谱，那你拍完就是比如说这是一个 red channel， 对不对？但实际上我们拿的是什么？零蓝样本，它是一个自发荧光，自发荧光是有不同的这个荧光寿命的这个长度的。所以这个时候如果我们打开这个荧光寿命 matrix 的话，会发现你看这颜色是不一样的。这个颜色并不是通过不同的 spectrum， 而是通过它本身不同的 fluorescent lifetime 而区分的，所以上的这个维彩。那这个东西一个是专门研究 fluorescent lifetime 的老师会比较感兴趣，还有它其他还有什么应用呢？就是说我们可以通过一个叫 phasor plot 的东西，这是一个小工具去做通道拆分，把不同颜色的这个通道拆分出来。那么实际应用，比如说我们像现在实现一个多通道的这个超分辨。那比如说这个样品现在用4个不同的抗体去标完了之后，染了 star red 和635 4种不同的2抗，但是实际上呢，我们只是怎么样？用了2种颜色，就是 red 跟635其实是一个颜色，然后 orange 跟580其实是一个光谱颜色。我们拍 confocal 看出来其实就2个 channel， 你看不出4个 channel 来。然后我们现在加了 STED 激光之后，当然它能够出现非常好的超分辨效果，但是还是2个通道，没法做多通道的超分辨。大家如果做过超分辨，肯定清楚，就是想做多通道的超分辨，其实实现是非常困难的。那么有了 timable， 我们可以实现一个4通道的超分辨，就是这个时候你打开 timable。因为 red 比635这个染料的 fluorescent lifetime 要长，那么它就可以把这2个光谱一样的染料，但是 lifetime 不一样，给它区分开。这边也是同样的情况，所以最后就出现4个颜色，这4个颜色都是 STED 超分辨的效果啊！你同时实现了4色超分辨。大家看 paper 的话，可以发现这样的这个结果是非常少的。那么想实现这样的一个效果，就是 flame 加 STED， 就 timable 加 STED。好，最后呢，再给大家总结一下，就是说 我刚才讲的是一个一个跟大家讲的这些 feature，但实际上这些东西是可以联合使用的。所以举最后一个例子就是，我们还是拍那个 carcassel 那个样本，那么我们现在 confocal 拍糊糊的，加了 STED 分辨率有一个显著的提升，对不对？那么我们又加了 matrix，就可以把背景去掉，看起来分辨率又高了，又通透了。然后这个时候我又可以加 timeball，把那些 fluorescent 更长的再去掉，把背景再去掉，进一步的去掉，因为 fluorescent lifetime 可以做这件事情，整个的图像就有一个显著的提升。当然这里边没有那个这个可变性镜，但实际上这些所有的功能都是可以放到一起共用的，所以可以有各种各样的 combination。好，就是这样，大家有什么问题吗
？OK，大家如果有谢谢天一博士，大家如果有什么问题的话，在这边可以随时提问。OK，Peggy，请说。那个许博士
听得到我的声音吗？
听得到，
听得到。
OK，我想问一下，那个你们说 STEDYCon 跟 facility 这两个不同的 modality，一个 resolution 是侧向，我现在只有说 XY，一个侧向的 resolution 是52奈米，另外一个是20到30奈米，所以我想确定一下，我们除了 power，就是在 facility 这个 modality 里面，除了 power 比较强之外，还有什么其他比较 tricky 的地方是可以让它的 resolution 增加的吗？
其实主要的就是STED laser power，这个是最重要的事情。
Okay，
然后另外一个就是其实 STEDYCon 它实现的那个多纳圈，它的那个样子的完美程度没有 facility 那么完美，所以它的分辨率稍微比facility 要稍微差一点。
但你现在说的都是 XY，就是 lateral 而已，对不对？
对。
那但是这样的 resolution 我们不需要再透过 deconvolution 就可以达到20到30奈米的 resolution。
对，完全不需要。就是如果这个机器您现场去看的话，我们拿一个壁子去拍，可以轻轻松松的可以到25纳米。
OK， 好，谢谢。
OK，看是不是还有其他的问题？老师请说。那个王老师，
我有一点就是前面的slide有点忘记，所以说如果我把那个 3D mode 把它开到极限的话，facility line 它的 XY 的 resolution 是可以到多少？
可以到75。
75，然后就是所以说
Z 方向也是75，
就是 XYZ 都是75这样。
对，所以叫各项同性 isotropic 的这样的 resolution。
OK， 了解，谢谢。
所以但是对不起，我自己也有一个问题，就是当我们在 XY 轴向的时候，其实我们也还是可以维持到25，没错嘛，对不对？只有再加上 Y， 如果我真的需要维持 isotropic 的话，那么我才去考虑到70个奈米的这一个。
对，这就是我们最大的优势，就是我们可以随便调，可以要 XY 分辨率极致，你也可以要 Z 的一个极致，也可以中间取舍，就是 XY 比25稍微差一点，30、40，但是 Z 方向你有一个80、100这样也都可以。
OK， 所以蛮活络的，就对。
对。
OK， 好，了解，跟我理解的一样。好，因为接下来我们会请那个 Dr. Gero 这边帮我们就是 online， 所以假设如果说有什么问题的话，还是一样随时可以提出。那么如果今天来不及问，或者是说不晓得从何问起的话，那可以联络那个我们就 Neil 或者是 George 我自己。老师请再说。
对，就是我突然想到，就是那所以它的 sample preparation 上面跟原本的 STEDYCon 上面有什么不一样吗？还是其实都是就是 sample 都是一样的？
其实 sample 是没有区别的，但是有一点其实要注意，就是我不知道大家平时做这个封片的时候，它封片剂是有区别的。有的封片剂呢，就是它并不在乎你整个3D 的 structure 它塌下来。因为它只拍是 XY， 只在乎 XY 的分辨率。这不只是对于 STED 技术啊，就是所有的样本制备里边，如果你想拍3维的结构，实际上是需要不同的 mounting medium 的。那所以如果你真的是想拍3D STED 的话，那一定要用可以维持它这个3D structure 的这种 mounting medium， 这样拍出来的结果才是真实的。否则的话，你整个拍出来的像一个 projection。
所以应该要用那种会完全 cure， 就是完全硬掉的那种吗？
其实完全硬掉的，像我们有这个 solid 和 liquid 其实是拍3D 用的，solid 是拍2D 用的。
那你们会推荐什么样的？像如果是 prolong 系列，我们就是还是用 prolong 系列嘛，或者是 prolong 系列有什么比较好？
prolong 系列是可以用的拍 STED， 但实际上他们我们这边得到的信息 prolong 他们就是 thermal 的那个东西，实际上不太区分这个。但是我们 Abberior 实际有自己的染料公司，我们也出染料，也出2抗，然后也有自己的这个封片剂。如果大家我不知道从George是不是拿到那个试用装那种染料，然后里边都带那个封片剂的，然后里边有 solid 和 liquid， 然后 liquid 就是拍3D 的 ，solid 就是拍2D。但实际上你在最开始做这个 sample preparation 的时候，你不必上来就特别在意这件事情，主要是要把你的结果拍好，所以你用 solid 的是非常容易，就像指甲油一样，放进去以后它干掉了，然后就拍就可以了。然后后面你发现这个样品你自己拍的没有问题了，2D 出的都图很好，你想拍3D， 这时候你换成 liquid 保存的时间长，但是制备起来你还要用指甲油什么给它封片，否则它会漏出来，这就比较麻烦。
了解，好，谢谢。
不客气。
好。Neil 这边如果说老师这边需要协助的话，马上再跟老师联络一下好吗？
好
，OK， 好，还有没有那个老师或者是同学们有问题？这边可以提问。Okay, then, well, so we finish our lecture session. Thanks for Tian-Yi 博士. Thank you very much. And later on, I'll let Gero handle the session. Gero is now in Göttingen with our handsome platform over there, which is Infinity Line, right?
Today it's actually a facility line, so it's a different microscope from last time.
Cool. Okay, so the screen is yours.
Okay, I'll give you a quick look at the microscope, maybe with this camera. Oh no, that's not going to reach. I'll use my camera on the laptop for 1 second. Let me see, maybe I can lift this. There. Okay, so this is the microscope here. So we have the an IX83 Olympus body, microscope body. We have the facility line microscope here, and we have at the bottom on the far end, this is where all the lasers are, the excitation lasers, and some of the detectors and all the controllers are involved. And below the table is the STED lasers. You can also see here the autofocus unit. So we have an autofocus which can be used with STED laser on. So at super resolution, we can still do a focus lock with the system. We also have a transmitted light detector on the top, and then multiple excitations lines, as Tian-Yi has mentioned. We have multiple objectives to choose from, and we have in this one STED laser. So this will give us super resolution with orange, red, and long Stokes shift dyes. There.
Excuse me, Gero, can you use the full screen? How about the full screen?
Full screen of the you would like me to share the screen now, Sisba?
Yeah.
Yeah, of course, I will. Yes, don't worry. Okay, I hope you can see now my screen.
Okay, that's far better. Yes.
Yes, so this is our Lightbox software interface, and it's meant to be as simple as possible to get super resolution images. So what we see here is a sample which is containing 3 stains here. I think there's also a DAPI stain, which I haven't been imaging in this one yet. And we can see here 3 colors. In green is actin skeleton, and then we have nuclear pores in magenta, and in the cyan we have the Golgi apparatus. Navigation is kept very easy here, so I just simply select an RI to select an area. And I can even turn these RIs here to give this a different layout. So let's go here. And then you can very easily see here the confocal images. And if I overlay them here quickly, we can see that as well. I can make custom overlays as well as I want to. Now the question is, how quickly can we get a super resolution image with this machine? For that, I shall zoom into, say, this area here. Bring this nicely into focus. I'm using my mouse wheel at the moment to focus. So I don't always have to go for the focus wheel here physically. And now I will switch on STED. Let's do this first for one color here, STED. And here you can see how this is basically adding one line here, symbolized by the donut. For the STED donut, we get the super resolution channel. And then we just press scan. And we can see here our confocal image on the left and the STED image for the star red channel on the right. Now you're wondering why what this number here is. Maybe this is the STED laser power, how much laser power we use with the STED laser to get super resolution. I think this might be interesting, so I'll deactivate these two channels here for now. And let's start with something like 1%. And then you can see we already get a slightly sharper image on the right. But if I go to 2%, with the STED laser power, increasing STED laser power, we get a better resolution. At 5%, we already have a much, much better resolution here. And what I was showing you was 10% resolution here. And now we can see these nuclear pores really one by one detailed. And what is also happening is that the pixel size is automatically adjusted, but you can also overwrite that here, no problem. So you can choose whichever pixel size you would like to, but you get a good suggestion by the software, making it easier to use. This here is two-dimensional super resolution. So now we're getting in X, Y, a super resolution. We can do the same thing with two channels. So I can reactivate here the orange channel and do super resolution with this orange dye as well. Let's have a look. And now we see a super resolution image for both channels, and we can do the overlays here, and we can see here the confocal and the STED images, so the confocal on the left and the STED on the right. And I think it's clear that you immediately, even at low laser power, you get already a very good resolution increase. I can increase this more, and then we should see here a even sharper image of the Golgi apparatus, if you want. There. So very easy to get a super resolution image here. Next step, I think I will focus on the nucleus here, because I would like to show you now what 3D super resolution is and how it works. So Tian-Yi has obviously explained already what 3D super resolution is, but I think it's very easy to show here as well with not a square ROI, but with a line ROI. So what I will do is I will cut through the nucleus like this with a line. I'll get rid of the other two channels. And now I switch on STED. And what you see is you get actually a very good resolution in the XY axis along this direction, but not so much in the direction Z. In Z, we still have the same resolution as in the confocal image. Even if I make the pixel size here in Z smaller on purpose, you will see still same resolution as confocal along the Z axis. What we do need is the Z slider here, and we go from 2D to 3D. I'll also make the pinhole a bit smaller. And this is all we need for 3D super resolution. So now we can see resolution in 3D, and we have here really, sharper images of the nuclear pores in X, Y, and Z. we can apply these settings. If we like these settings here from the 3D super resolution, we can apply them easily to a section of our sample in a volume scan, because this is where this becomes interesting. So let's say I go here, place an RI here. As I said before, I can even turn these RIs as well. And then to create a volume, I only have to go live, scroll till I am at one end of the structure, and then up to the other end of the structure. And this is defined my volume here. I can very easily now apply the settings which I created here by just using drag and drop here, then pressing scan, and that should easily now get this going. While we're scanning, I can switch to a 3D view as well, and I can see my image coming up during the scan here. Here, also go to the volume view, and we can see this is the confocal image where you can't really see the two layers of the nucleus very easy, not so easily. Why? Because the resolution in Z is just too small. But when I use the 3D STED, we can easily see this is a kind of taco shape like this, and we can turn this here. We can also go and do some rendering if we want to. We can do an export of this animation. For example, I can say I want to turn this in the y-axis, this image, and I want to have here a bounding rectangle as well. And maybe I'll put this just to the desktop, make a new folder here, demo. And if I export this, I will just simply get a small movie there, which you can then So it's a very fresh computer here, so need to start every software. So now you can see you have here a little animation, which might be quite nice if you just want to present your results in a PowerPoint presentation, for example. Now I can take this a little bit further, and I can go for a higher resolution. Now we've seen that at higher resolution we get some smaller pixels. At higher STED laser power, we get a better resolution here. So let's say I go here for 30% STED laser power. Then we can see here, we get an image with much higher resolution here. Now, how would I put together an image here with adaptive illumination? So the trick is with adaptive illumination that we can say I use 20% here, and I switch this on. I get this little option here. Let's start with one step. And what we can see here is a mask. And this adaptive illumination, which I would like to show now, is really there to reduce illumination of our sample. We call this feature now flexposure because it's a flexible exposure of the sample, meaning we can adjust how much of the sample is being illuminated. And we can see here with the mask which part is illuminated. So everywhere where the gray mask is, there's no sample illumination. How does the machine decide? Well, it tests here in the confocal channel. Do we have signal? And if no, in the dark areas, it will apply this mask. The threshold for the mask you can see here. It's very easily adjusted by the user. And that means that only the structures which are fluorescent are being illuminated, and the dark areas where we have no structures don't need to be illuminated and will not be illuminated with a STED laser in the second step. Now we also have this two-step here. Oops. And if I adjust this parameter here, we will get very much reduced illumination because we now have this second gray area here. So if I overlay this, you can see in the STED image, there's most pixels here are not illuminated. They are all grayed out. And there's only a very small area of pixels which is actually illuminated here in the end. If I now go back to my sample, we can have a look and do another volume scan. So let's say I go and produce a volume here. Let's say in this area. Go live. Select a slice below and a slice above. And now I have a volume defined. And now I take the settings from here with adaptive illumination. So flex exposure is on. And we just scan. And we see again a good 3D super resolution image here of our nuclear pores. Once the scan is stopped, we can check here. And we barely have any bleaching. So I think it's very hard to see. There's maybe a little bit, tiny bit of bleaching in this RI here, but not much. Now let's repeat the same settings here in the next bit. And this time we again select our top and bottom slices here. Yeah. And we take the same settings as in our previous volume image here. However, we will switch off the flex exposure. Now we get again a stack. And while we're recording, we can see the stack being assembled. We can see also confocal versus STED here. And much higher resolution in the 3D super resolution image here. And once the complete thickness of the nucleus has been recorded, we can switch off the acquisition here. And we have another look at our nucleus. And you can see there's a lot more bleaching in this area. It's a lot darker here. And that means we have illuminated the sample much more in this area here. We zoom in. So we can see here, this is getting darker in there. And the more, the higher the resolution is that I'm using, the more bleaching I will get. And adaptive illumination can really reduce that bleaching effect. In the next step, I would like to show you a little sample where we have actually even more interesting structures to see. So very shortly, we'll change my sample. And while I'm changing my sample, if you like, you can always ask questions during this process. Samples are similar to any confocal samples, simple slice with a cover slip on top, and we'll have a look at that. Second. Okay, so now we have a similar sample. However, what is different is that we have not a central part of the ring. So in the last images, we were seeing basically always a just dots here. That was because we had marked the center of the nuclear pore. So the question is now, if I use a sample where we have marked somewhere parts of the ring structure, what will we see there? For that, I will just simply now start scanning in the new sample. And we'll have to see if we can find a good nucleus here. We don't have any of the other stains here. It's a simple red stain. Let's have a look. There's the cover slip surface. Let's see if we can find some nuclei. There we go. And you can see how I navigate here with my mouse. I'm just dragging things here with my mouse into position. So if I want to move this to the side, I can do so. And I can move this also into the center for imaging. And my mouse wheel is, as before, my focus wheel. So now I'm perfectly in focus. Maybe use 1% excitation here. There we go. Next step, I'll use an RI to zoom in onto one of the nuclei. And now I will try some super resolution imaging here. So I simply switch on STED with, say, 10% laser power, make the pixels a bit smaller. Well, let's actually start here with 5%. Why not? Really low. And what we see at 5% is already that here we might have something different from just a normal, or let's have this at a little bit more accumulations. You can already start seeing that these are not entirely dot-like structures, but that there's some substructure to this. Now we cannot discern entirely yet that these are rings, but how about when we use deconvolution? Now we have here a processing menu already in here, but in the upcoming version, we are having a new deconvolution software, and that is called, or new deconvolution feature called TrueSharp. And that's much improved over the previous one, which you still see here in this software, in the sense that it can remove background, out of focus light, and artifacts really, really well, while still getting a very high, much of a gain in resolution. For that purpose, just to use the new feature, I'll save this image here as a copy. Go to the demo. We call this NPC STED 5%. And then I will start the Lightbox version, which has the deconvolution feature in it, this one here. And I'll simply drag and drop that image in here. And deconvolution is easy. I'll simply drag this in the processing area. Add plus here for deconvolution. And here we have the iterations of deconvolution. We have background options. This is what is new. And this helps us to suppress artifacts and out of focus light. And we have here our PSF options. If I then go run, I can see here the deconvolved result. And if I make this, let's say here sharper, there, and now we can actually see this already quite well as ring-like structures. So I think the deconvolution feature really adds image quality and resolution in this case. And what it also doesn't do is amplify artifacts in the background. What do I mean by this? Well, let's switch off this background reduction. So if I switch this off, you will see actually all these little dot-like structures amplified here in the background. So this is, I think, the big advantage of this version, that we really here have this background subtraction option, and now we really get just exactly what we need to see, which is the real signal amplified and sharpened. Let's go back to the software here. Obviously, now we've done this image here at a very low resolution. Let's try this again at a slightly higher resolution. Let's see if we're still in focus here. Let's go change this maybe to this layer. Here. Switch that on at 10% power this time. And now we can already see these rings a lot more easily. Let's try the same at 20%, and you will see that this is very easily visible here as rings. In fact, now the resolution is so high, we might as well want to do this. And I'll go here for a different region. Let's see this one. Oops. There. And I will instead this time here make the pixels still a little bit smaller than that. Let's have a look now. And now we can really appreciate, I think, the ring structures, and we start seeing some substructures here as well, subunits. And again here, I can now export this one, MPC. I think this was 20%, step 20. Yeah. And I'll go to our new software here, drag and drop this image in there, and do a deconvolution. Again, just a low background subtraction because there's not so much background. And we can now see an image here where we can see, oh, maybe I can make it a little smoother here. Let's try that. Yeah. And I think we can very well see that we have here single subunits, which we can see not all subunits are labeled, but in some there's one, say 4, 1, 2, 3, 4, 5, and so on subunits in there. So I think down there is a few nicer rings here. You can see here the individual subunits already. And we're working just at 20% of the resolution, of the maximum power of our STED laser. So in the next generation of the acquisition software, the deconvolution feature will also be directly available while imaging. We don't have to then drag and drop this into the new version. This is just the current version which is delivered, and the other version software I was showing you is the one which is the next update coming to all customers. That was the TrueSharp feature, which can really enhance our images. But keep in mind, every time I was imaging here, before I press deconvolution, this is all completely raw data. So the image here is showing for each pixel how many photons have arrived in that pixel without any subtraction or sharpening or smoothing. So this is the absolute raw data, raw image here. So it's nothing has been changed in this image. Only when I was using deconvolution, then I was making any changes, any post-processing to the image. So we typically show you completely raw data, but we have nice new deconvolution features with TrueSharp. And the next thing what I would like to show you is a feature called Timebo. And Timebo is our lifetime imaging feature. For that, I will unfortunately have to switch the sample one more time because we need something which shows different fluorescence lifetimes. Let's have a look. In the next sample, we have an orange stain, but it's not only one orange, it's two oranges. So basically we have two dyes, both with the same color. Spectrally, they're not really separable, but they have a very different fluorescence lifetime. For that, I will simply go to the new sample, make a new sample, and you can see our software always starts with the dyes. You don't have to go and select a lot of different settings and so on. What you basically tell the software initially is which dyes do you have in there. And I'll just tell it we have an orange dye in here. And we can go to call this sample 2 color flim. If I want, I can give this also a description here, and I can say this is star orange and CF594. That's the dye we are looking at. I can also choose my overlay colors here, and I might just go here for grayscale for the start. I'll do a new overview, and I go live. And what we see here is again nuclei. Maybe I crank the power a little bit up. Yeah, that's better. And we see nuclei with a labeling also in the Golgi, for the Golgi apparatus. Now, the next step will be that I'll zoom in onto one of these nuclei here. And I'll zoom in a little bit further yet to see some more of these structures here. Now what we see here is basically a normal confocal image. And what I would like to show first is how we can sharpen this image a little bit. So this is going to be a combination of two features I'm going to show. There's the matrix array type detection, which Tian-Yi has introduced. We are looking here not at a point detector, but an array type detector, as in other microscopes also seen, but we are using ours here also for lifetime imaging and for super resolution imaging. So let's have a look. What would the matrix detector do in this case? When I activate matrix here, I get automatically a larger pinhole. I will also crank up the accumulations here a little bit. Let's have a look. Now it's changing the spectral detectors to divert the orange light onto the matrix detector. Bring this back into focus nicely here. Say here. And what we can do is maybe I'll get a little bit more signal. Say there. Yeah, that's better. What I can do now is I can do processing, post-processing. So because with an array type detector, we can detect the in focus and out of focus light physically and physically separate the in focus light and out of focus light. And that means I can use here a differential detection function. Whoops, sorry. Yeah. There. And that will basically just sharpen the image here by taking out the out of focus light of the brighter areas. What it also allows me is to do some additional sharpening functions here, and then we really get a better resolved image, even in confocal mode here. And that will especially be true also in the Z-axis. So we get a much better sectioning, less background in this image. So when we compare this to our confocal image here, and we have here our sharpened image. Maybe let's give this the same color scale. So you can see really the resolution does go up from confocal here to our matrix image here, where we get a better resolution and also less background. However, the next thing the matrix detector can do is lifetime imaging. We can also do this with our normal detectors. However, with the matrix detector, the lifetime imaging is directly built in, and it can be done at an even higher time resolution. And as you have just seen, we can also sharpen the image in confocal mode here with the matrix detector. So what I will do next is I will basically switch on lifetime imaging. We call lifetime imaging in our machine timebo, and this can be seen here in this little timebo menu. I simply switch on timebo. And give this a longer time for acquisition. I stay into focus here. That's a nice section. And we still see a grayscale image. As before, I can go and do some processing to sharpen the image a little bit. This is the matrix processing. And then I can give this actually now a different lookup table, because we have now lifetime information. And I can do this here. And we can now see color-coded the lifetime. So if I go here from 1 to 3 nanoseconds in the lookup table, we can see actually there's two very different lifetimes in this. And we can see that the nuclear pores are labeled with a long lifetime dye, which is our star orange. And we can see here the short lifetime, which is our is CF594 from Biotium. So two different lifetimes. What we can also do is we can now create a phasor plot. And what you see in the phasor plot is for every pixel in the image, we get a little dot in the phasor plot. That dot is placed in the phasor plot according to its lifetime. And pure lifetimes will end up roughly on the semicircle or half circle we can see here. Short lifetimes will end up here to the very right, and longer lifetimes will end up on the towards the left. So we see here a fraction with about 1.7 nanoseconds, which is exactly what we expect for CF594, the Golgi stain here. And we have one around 3 nanoseconds, which is our star orange dye. All this has happened without me using any parameters or special settings. So I don't need to know anything about exponential fitting here. I simply get a graphic representation of the two lifetime components here in this phasor plot. The interesting part is I can also now go to the phasor plot and select one of these populations. For that, I'll just take here a circular RI or ROI, draw this, and I can select this fraction. This will be just my CF594 stain with a shorter lifetime, or I can go to this one here. The other thing I can do is I can use a line tool here and create a gradient from one fraction to the other, and there we'll have a very smooth separation of the two. And I can also get mixtures, which would be anything which is here in the middle of these two. I guess another question is, can we reproduce this result here in with STED? So for that, let's go here in nice and focus there. And now I switch on STED. And I will activate Timebo for the normal confocal channel and the STED channel. So it's as simple as that. I have here my STED channel, Timebo activated. I will also give this a little bit higher excitation and smaller pixels since we have a higher resolution. Now you can see on the left the confocal image, on the right the STED image, where we can see all our individual nuclear pores. I'll get rid here of the confocal image. And here for the STED image, I will now go first and do the matrix processing. And you can see that the background signal has been removed strongly. So now we have a slightly sharper or less background in our STED image. And now I can take again a look at the lifetime, and we can see here our lifetimes again, and we can create a phasor plot and separate the two fractions again. And now we have a super resolution image of the two dyes, and they can be separated via this line tool very easily in the Timebo module here. So we've seen two modules here. One is matrix, which can remove the background. So if we go back to our images here, we had a confocal image versus a processed matrix image, which was a lot sharper in the end. And the matrix straight away allows you to do Timebo imaging. So this is when you get a license for Timebo, you can use the matrix detector straight away for a lifetime imaging. You can also use the APDs that will then require a hardware upgrade to enable all the APDs in the system as well for APD detectors for lifetime imaging. So two modules, matrix for sharpening is an array type detector, and we have Timebo for lifetime imaging. After this, I would like to switch to another module we have, and this is the adaptive optics module called RayShape. Tian-Yi has also introduced that one before. And for that, to show that module, I will simply show you a sample which is much deeper than this. So this sample here was just cultured cells, and they were labeled, but they are very thin section on it, very thin layer of cells. I'll now use a sample which uses the same mounting medium, the mounting medium we use for nearly all of our samples, which is called Mount Solid. Abberior Mount Solid, very similar to Prolong Gold from Invitrogen, for example, or Thermo Fisher. So standard mounting medium, but a very thick layer of it. And we put some beads with a defined size in there and a defined brightness. So we can have a look what happens when we go deeper into the sample with normal mounting medium. Okay, let's have a look. I shall close the lifetime sample here. And this one as well. And I'll go to new sample. And it's simply a red dye in there. So the beads are stained with a red dye. Call the sample bead 3D layer. And I will just use first my focus wheel to get roughly into focus here. And we should soon see some beads. Here they are. So very dense layer of beads in here. What I will do now is I will do a vertical scan through the sample. For that, I'm using not a square ROY, but a line ROY. And I'll place it just here somewhere in this area. Now, when I scan, I can see here our beads and on the top, the glass layer. What I will do is I will make the pixel size a little bit smaller so we can see the shape a little bit more. Now we can see the beads, even though they are 120 nanometer sized small beads, appear, well, kind of oval shaped or egg shaped in the Z-axis, which is normal because this is a confocal resolution. What I will do now is I will go further deeper into the sample, and there should be a very, very thick layer of beads. So we can see, yes, we can go much deeper. I can also just go here deeper and deeper and deeper using this Z tool here. Let's say we go 70 micron deep. And we can see now at the top, we have a very nice image, and the bottom, it gets darker and everything gets very much stretched out in the Z-axis. So if I zoom in here, we can see here, everything is stretched out and dimmer. And here on the top, everything is much sharper. How can I remedy this? Well, I can go and use adaptive optics, which is our reshape module. I can just open this dialog here and switch this on. I have now two controls, one slider for the top of the image and one slider for the bottom. At the top of the image, everything is fine. The aberrations occur more and more the deeper I go into the sample. So I have a slider for the bottom of my image here. And there I will actually make a change, and I will apply some adaptive optics there, some spherical compensation. And you can see now I've actually restored the quality. So going from the top to the bottom, I have pretty much the same shape of the PSF here and here, while previously I had at the bottom this stretched out and dimmer PSF. Now this is especially interesting when it comes to 3D STED. So let's repeat this image here. And this time I switch on STED and I switch it on in 3D. And you can see we can get a 3D super resolution image all the way to the bottom here. Adjust the lookup table here. You can see the beads in the proper size and shape. And we can see this all the way to the bottom here. Now, if I repeat this without the ray shape module on, no adaptive optics, we can see that the image deteriorates the deeper we go into the sample. So we can see here, all the way to the bottom, a good 3D super resolution image, and here, very much a deteriorating image quality, especially to the bottom. So here we get mostly background and not any signal in there. So the signal goes down a lot. In the confocal again, we have then a smeared out PSF towards the bottom. Another thing we can try here is maybe the matrix detection. And we can now see with the matrix detection and this background subtraction, oops, here, we get a much better signal to background than here before. And so here with a normal pinhole at 1 AU, well, we could make the pinhole a little bit smaller, but we get a little bit of background in our image. And here with the matrix, we get a much reduced background signal in our images. So the matrix detector can be used here also on top of everything. So the first thing we're doing here is we're using ray shape, the adaptive optics module, to compensate aberrations which occur when we go deeper into a sample. I think it's very clear that this doesn't really happen at the top of the image or very near the cover slip, but when we go deeper in the sample, we do get worse image quality, especially in 3D STED. We can see this here, but with the ray shape, we really recover it. And then if we add our array type detection on top, we can really get rid of the out of focus light as well. Okay, so this was just a vertical slice. Obviously, this is more interesting when we go for larger structures or deeper into the sample and we want to navigate to new structures. How would we do this? So I'll go to my overview. And then I use my slider here to go deep, to say 50 micron deep into my sample. I make a little square, Roy. Say this is the start of my Z stack, and I go a few micron deeper, and that's my Z stack defined. And then I can apply, I don't have to figure out how much compensation I need to do with my adaptive optics. I just transfer the settings here, and it's automatically applied here. If I then go to scan, we can see our image here live. We can go, for example, to volume mode, have a look at that. And then if I switch off the adaptive optics here, we can see the image quality drops. So you can see immediately here, we have basically lost our image quality, very little signal. If I switch it back on, you can see the signal coming back as well. Yes, at this point, what I would like to do is maybe go and recap quickly what we've done. So we've looked at super resolution in 2D and in 3 dimensions. We've seen that when we increase our STED laser power, we get better or higher resolution. And we have actually a lot of reserves with this high power STED laser because we were using only, I think, 20 or 30% at the maximum in our samples there. So much reserves for even higher resolution there. We were doing this in 3D. We were doing rendering. We used also our new TrueSharp deconvolution module, which helped us to discern these little ring structures in nuclear pores much more easily at lower STED laser power even, and then improve the image. But at higher STED power, again, we got more resolution and we could see even the individual subunits or parts like of the ring shapes. We then went on to do lifetime imaging, which we call timebow in our machine. And we combined this with the matrix array type detection, which sharpens the image and it has a very good time resolution, which is good for lifetime imaging. And then in the lifetime module, we were separating 2 dyes with different lifetimes very easily using graphical tools, using the phasor plot tools. At the end, we now went to have a look at the adaptive optics and how we can use the adaptive optics to go deeper into a sample and get a good image quality deep in a sample. If you have the time, I could have quickly show you how this might look in a larger sample, say a Drosophila stained Drosophila sample, if you like. Just let me know if you would like to see this, or if you would straight away like to discuss the results of what we've seen today.
Yes, we should go for the other demonstration, please.
Okay.
So in the meantime, if we have anything, any question to talk, this is the time. We have short time. If there's any, just let me know. Yes, Wang Siqing. Yes.
So I have this question about how the usage of this phaser or the time thing. So for example, when we are do the spectral imaging with TIES or Leica, so normally you need to generate this reference by yourself, recording the whole spectral of your own dye or so. So with the phaser or this time thing, do we need to record or have our own reference for that? Or if we just use the same fluorophore or the dye as you do, we will automatically eligible to use this reference as you do.
That's a very good question. So what I've been doing is I was basically doing it as if I don't know what is in there. And I was using the phasor plot to see the 2 populations, and then I can separate them. If you have a dye with a known lifetime, you can also directly use it. And there's something which we call a split mode. And in the split mode, basically I can add 2 dyes with different lifetimes, and it automatically does this line tool for you because we already know where this needs to be placed. So it's basically without even seeing that, it will just generate 2 channels and separate these things out. Obviously, if you have a new dye, this is very useful that you can use the phasor plot. Have a look, what is your actual lifetime in there? Also, the lifetime might change with a different mounting medium, so that would help. And in that case, we would have to, we can then generate also a custom dye in the software. So that's also possible that you go and if you take a new sample, you can go and you can go and customize a dye. So you can go here, clone and customize, and you can make a different dye and change the spectrum if you want. And you can also provide a different lifetime to the dye and so on. And then you have a new dye and it will automatically then take these settings to do the split in there.
So if say we have a dye that have never been tested before either by us or you, so it's just be on the safe side to check its lifetime.
Yes, yes. And I find that many dyes do have a different lifetime when they are used, for example, in a very dense labeling or in a different mounting medium. So okay. This can also happen. Also, it can be very useful to identify, say, background fluorescence. So if you have something like a, let's say, autofluorescence there, which has a short lifetime usually or very often, and then you can differentiate that, identify maybe in the phasor plot where it is, and then separate that out as well.
Okay,
if it's
thank you very much.
Okay, let me just quickly check that we are on the structures here. I think so. Okay. So here we have some structures already. As I can see, it's actually very bright in the red channel, so I might just lower my excitation power there a little bit. And I use this now as a big overlay. In blue, we see actually trachea and so on, which are typically giving a lot of autofluorescence here. So we can see these in the DAPI channel. It's not actually a DAPI staining in this case. We see some what's there, and in the orange, I think the orange is a little bit weaker. That's a slightly older DNA stain here. And now I'll just navigate this a little bit to see what we have here. That's not the part I wanted to look at. Aha, here we get a little bit more interesting parts. There we go. And what I can do now is, if I want to have a larger view of this, I can make a new overview with a large range. And I can let this run as far as I want there. So now it's going to do a tiling. And you can see how it's now going to do a larger image here. And it's going to go in a spiral pattern until it has captured the whole range, which I have provided at the start. So I was saying do 1 millimeter by 1 millimeter. If you like and want to do this faster, you can also switch to a lower magnification objective lens. That's also possible. I think I might have a look here in one of these areas down there. So I'll just use so stop the acquisition here, and I'll zoom a little bit further down here. Let's have a look what we can see in that area. Okay, here we're getting a little bit brighter staining in the, okay. This might be getting a little bit more interesting here. Let's have a look. Let's do a proper overview here. So here we see trachea again. So these are Drosophila organs kind of squashed on the between the cover slip and the slide. And maybe I'll just zoom in somewhere here now. Let's say I'll zoom somewhere in this area. And the first thing I can do is I can do a vertical slice through this. And have a look in the Z-axis. You have to go quite a bit further up here. And here we can see the top of our Drosophila organs, and then we can go a little bit deeper here again. There we go. Now the question is, does our image quality stay the same, or does it also get a little bit worse when we go towards the bottom of the structures? And it does seem to smear out a little bit, but not too terribly bad. So now we can try maybe some 3D super resolution in here. So I'll get rid of the other structures here. Use just the red channel. And let's have a look. That's 2D super resolution. And we switch to 3D. And we can see we get already quite deep into this because it's mounted in a very good mounting medium. But towards the bottom here, the image quality gets quite a bit. It's worse. So you can see here the 3D super resolution image. You can still get a little bit sharper image here than in the confocal image, but not quite as sharp. So what I can try to do is to use some adaptive optics and compensate a little bit when we go into the deeper parts. And I think now we can see that here in the deeper areas, we now still have a nice and crisp image. So you can see here, these structures here, nicely resolved, much sharper than here in the normal confocal image. And when we compare this here to when we have no adaptive optics, here it's much more background and less sharp on the bottom. So I think maybe this way I can maybe show them more easily overlaid here without adaptive optics and here with. So without getting blurrier and hazier on the bottom, and here much sharper, high contrast here in the bottom areas as well. So yeah, even in a proper Drosophila sample here, the adaptive optics really makes a difference. Here versus here with all the detail. I hope this was an okay introduction to some proper imaging with the microscope. If you want to see anything else on this Drosophila organs, feel free to ask me. I can take some more images. Just wanted to show that we can get a nicer image quality here, going deeper into the sample with the adaptive optics in a large sample as well. So that, for example, the 3D super resolution can be improved going deeper into the sample. The navigation is also doable then, even if you have a larger sample, you can either go switch to the eyepiece one way, but I unfortunately cannot show you what I'm seeing in the eyepiece. You can navigate using the mouse and the overview. You can do the tiling overview, and you can also switch, if you like, to a lower magnification lens and then do a faster, even larger overview. But the nice thing is you can always switch back, and you have always your R eyes and can go back to what you were doing. So for example, here in this sample, when I go back to my overview, I can see where my line was, where I was scanning vertically, and that would be here, this area down there. And yep. So I always know where my images had been taken, because I can always look at here my tree view and this structure here. I can also go and export this data very easily as either raw data for further analysis in other programs, or as an image, say an overlay here. And I can just export this file. And maybe I select actually here this on the desktop. And then you have a nice image with a scale bar and so on, multiple options. You can even do a batch export, say export all the images in here and select what you want to do with them, which overlays and so on you want to have. So very easy figure generation also in this software. Okay, I think at that point I would like to stop. I would like to know if you have seen everything you wanted to see.
Okay, thank you. I'm going a little bit with Mandarin. 老师同学如果说有什么问题的话，现在 Gero 在这边，那我们可以对他发问。那现在 symbol 上面放的是那个 Drosophila， 那所以上面如果要看别的地方，他可以帮我们再移过去看看，会有更多image 大家想要看的。好，那我简单讲一下，就是说，因为今天我们整个过程都有录影，所以如果说比如说同学们觉得老师今天 PI 来不及过来的话，那么我们可以找我或找 Neil 要这些连结，我们会把它给大家。那第二个是因为大家可能会觉得说，哇，资讯量怎么那么大哈？那其实 Abberior 已经发展了好久，他们都有自己的 building， 很漂亮的一个 building。那么我们今天在这边看很多这个 module 啊，什么都是有它的原因存在。但是我总是感觉说，因为我们比较晚才引进这一个设备，所以我们常常会觉得说，我们好像追剧追到季末了才回头看一堆东西这样子。我就觉得说，哇，剧情好多。那事实上它都有它每一个作用。简单的来讲，今天我帮大家 run through 一下，就是一开始我们了解了 STED 的基本原理。那我们想要看到 2D 的 STED 以后，我们希望把它 apply 到 3D STED。那在 3D STED 上面，我们必须要注意，在 2D 的 STED 上面，我们给了很多个 photon toxicity over pulse laser。但是现在我们讨论到 3D 的话，我们就必须要注意到它是一层一层一层的。那在这个过程里面，光路经过的过程里面，都会有photon bleaching 的现象。所以 Abberior 花了很多时间去第一个降低这些 photon toxicity over sample， 那这是第一个。那所以第二个，我们又要去解决掉在 Z 轴 Z resolution 上面，如果发到深的地方的时候，在别人都完全没看到的状况下面，那我们要怎么去做？于是他们用了最新的，我们实际上如果再回溯2年，我们想都不敢想会用 adaptive illumination 这样的技术。等于是非线性的这种镜头来做这些事情，但是都做到了，因为这全部都是最先端的东西，所以资讯量确实有点大。那这些 module 呢，有一些并不一定就一定要装。那回过头来讲 facility line 为什么叫 facility line？ 一开始瞄准的就是大概不会是只针对特定实验室去设计这样的产品。我们希望能够在这一个facility line 架设的时候，最好是架设在 core facility。那么 core facility 服务的对象比较多，那所以我们就必须要具备各种可能要看组织的，比如说 Drosophila， 那比如说想要看细胞 sample， 那就会有不同的 module 去对应。那么特别今天我们第一次看到，我们在 categorize 各个 channel 的时候，一般都是用波长，然后用很多的镜片去反射，我们去 categorize 这些不同波长，然后我们染上不同的颜色。但是今天我们大家都学到的就是，原来时间轴的 resolution 可以用到，所以我们现在变成 x、y、z、t 还有 lambda， 所以这个都是我们可以去 categorize for instance channel， 所以看到真的是很多的东西，也许有一些事情，碍于时间上的关系，其实我们今天准备的也蛮长的，碍于时间关系，我们没有办法讲得很清楚，因为资讯量真的很大，跟我讲的一样，我们是在季末的时候开始来追剧的。那我们今天也请到天一博士他是比较常住在北京。那么 Abberior 这边我们有很多个application engineer、application specialist。我想我们最大的好处是，我们都非常喜欢谈这些 application 的东西。但是如果要谈到这些 opticals、engineering、technical science 这些东西，我们都有很多对应的人可以来跟各位解释这些东西。Okay, so if no further ado, anything to add from Dr. Gero? It's okay, right?
Okay. All right.
Okay. And then Tian-Yi, anything to add?
您已经说的非常好了，感谢。
Okay, then thank you very much. Then
谢谢大家。
do remember to let us know if you have any further question. We are more than happy to answer your question. And if you have anything to contact with, for example, to Gero, I will collect all those questions, go to Tian-Yi and go to Gero. I promise that I will look after them to give you a satisfied answer. Okay. Thank you very much.
You.
Thank you. Bye-bye.
Thank you.
Thank
Bye.
you. Bye-bye.